Welcome to G Fun Facts Online. Imagine descending into the ocean, leaving the sunlit surface far behind you. As you plunge deeper, the vibrant blues and sparkling reflections fade into a heavy, monochrome blue-black. By the time you reach depths of 20 to 200 meters, you have entered the ocean twilight zone, also known as the mesopelagic realm. Down here, sunlight is a scarce currency. Colors disappear, shadows soften into a perpetual, gloomy haze, and the ability to detect the faintest glimmer of light can mean the difference between finding a meal and becoming one. For more than a century and a half, biology textbooks have painted a very specific, rigid picture of how animals, humans included, navigate light and dark. This 150-year-old rulebook dictated that vertebrate vision relies on a strict dichotomy of two cellular photoreceptors known as rods and cones. To appreciate the sheer weirdness of what we are about to discuss today, we first need to completely understand the biological laws it breaks. The retina, which is the sensory membrane lining the back of the vertebrate eye, is an astonishing piece of biological engineering. Its primary job is to catch light particles, called photons, and convert them into electrical signals that the brain can process into an image. Since the mid-19th century, scientists have classified the photoreceptors tasked with catching these photons into two neat, distinct boxes based on their shape and function. First, we have rod cells, the low-light specialists. Rods are long, cylindrical cells packed with rhodopsin, a light-sensitive protein. Their elongated shape makes them excellent photon traps. They are so incredibly sensitive that a fully dark-adapted human rod cell can respond to a single photon of light. However, this extreme sensitivity comes with a massive trade-off. Rods are easily overwhelmed in bright conditions. This is exactly why you are temporarily blinded when stepping out of a dark movie theater into the bright sunlight. Furthermore, rods do not process color, and they pool their signals together before sending them to the brain, which sacrifices visual sharpness for sensitivity, resulting in a blurry black and white image. On the other side of the dichotomy, we have cone cells, the high-definition color processors. Cones, true to their name, have a more tapered, conical shape. They require significantly more light to activate. However, they contain different types of photopigments called opsins that react to different wavelengths of light, allowing for the perception of rich color. Furthermore, cones often have a one-to-one connection with the optic nerve pathways, providing sharp, high-resolution, dynamic vision. For 150 years, this division of labor was considered an unbreakable fundamental law of vertebrate biology. The rods and cones slowly change position inside the retina when moving between dim and bright conditions, which explains why our eyes take time to adjust when we flick on the light switch on our way to the restroom at night. But the deep ocean has a habit of making a complete mockery of our terrestrial textbooks. In the twilight zone, this neat dichotomy falls apart entirely. The environment is neither bright enough for cones to function properly, nor dark enough for rods to be completely comfortable. It is a realm of perpetual, murky half-light, and nature absolutely abhors a vacuum of efficiency. This brings us to a revolutionary visual secret recently uncovered by an international team of researchers. In a groundbreaking 2026 study published in Science Advances, Dr. Lily Fogg, Dr. Fabio Cortesi, and their colleagues from institutions ranging from the University of Queensland to the University of Helsinki made a discovery that literally rewrites the rules of evolutionary biology. By examining the microscopic eyes of larval deep-sea fish, they found a completely new type of light-sensing cell. It is a hybrid photoreceptor, a biological chimera that physically looks like a rod, but operates using the genetic and molecular machinery of a cone. To truly understand the magnitude of this discovery, we must dive into the evolutionary pressure cooker of the deep sea. The mesopelagic zone, spanning from about 200 to 1,000 meters deep, is the largest continuous habitat on planet Earth. But the fish in this recent study were examined in their larval stages, captured at depths between 20 and 200 meters in the Red Sea. This upper twilight region is a highly active biological corridor where the physics of light behaves very strangely. Water rapidly absorbs the red, orange, and yellow wavelengths of sunlight. By the time you reach 50 meters, the world is bathed in a monochromatic blue-green hue. For the tiny larvae of deep-sea fish, this environment is a brutal, unforgiving obstacle course. They are only about half a centimeter long, with eyes smaller than a millimeter. They need to spot the microscopic zooplankton they feed on, while simultaneously avoiding the looming silhouettes of massive predators swimming below them. Furthermore, many creatures in this zone use a brilliant evolutionary tactic called counter-illumination. Because the only natural light comes from the sun directly above, a fish swimming higher up casts a dark silhouette against the faint, downwelling light. To hide this silhouette, deep-sea fish have evolved rows of photophores, which are tiny light-emitting organs on their bellies. By producing a faint, blue-green bioluminescent glow that perfectly matches the intensity and color of the sunlight filtering down from above, they effectively render themselves invisible to predators lurking below. To survive this high-stakes game of optical hide-and-seek, visual systems had to adapt dramatically. Neither standard rods nor standard cones were up to the task. Standard rods would blur the fine details needed to spot the subtle break in a predator counter-illumination camouflage, while standard cones simply would not activate in the gloom. The brilliant solution nature found was a biological mashup. During a series of marine exploration voyages in the Red Sea, the research team painstakingly collected larval specimens of 3 distinct deep-sea fish species. First is the hatchetfish, a bizarre-looking fish that, in adulthood, resembles a shiny silver coin with a perpetually frowning face. Second is the lightfish, a slender, elongated fish heavily decorated with bioluminescent photophores. And third is the lanternfish, one of the most abundant vertebrates on the planet, known for migrating up to the surface at night to feed and retreating to the depths by day. These fish are tiny, measuring just 1 to 3 inches as adults, and their larvae are little more than translucent slivers of life. Studying them presented an immense technical challenge. Because their eyes are smaller than a single millimeter, traditional dissection and cellular analysis require immense, painstaking precision. Using advanced genomic sequencing and high-resolution microscopy, the scientists peered into the retinas of these larvae and found something that simply should not exist according to the old rulebooks. What the researchers observed under the microscope was a cell that entirely defied categorization. They found that, as larvae, these deep-sea fish mostly use a mix-and-match type of hybrid photoreceptor. These cells look exactly like rods. They are long, cylindrical, and perfectly optimized to catch as many light particles as possible. But incredibly, they use the molecular machinery of cones, switching on genes usually found only in cones. Let us break down the absolute genius of this evolutionary design by looking at it like a computer system. First, we look at the hardware, which is the rod structure. To see anything in the twilight zone, you need a massive surface area to catch sparse, rare photons. The hybrid cell heavily borrows the physical architecture of a rod. It is elongated and cylindrical, packed end-to-end with membrane discs that act like a towering stack of solar panels. This ensures that even the faintest glimmer of downwelling sunlight or a brief flash of bioluminescence is captured. Next, we look at the software, which is the cone genetics. While catching the light is incredibly important, processing it quickly and sharply is equally vital to survival. Standard rods are notoriously slow to reset, and because they pool their signals together, they create a blurry image. But by utilizing the genetic code and molecular machinery of a cone, this hybrid cell can process the captured photons with the incredible speed, efficiency, and potentially the sharpness of a daytime visual system. This hybrid cell essentially takes the absolute best bits of both the bright light and dark light systems to become something entirely new that is perfectly optimized for twilight vision. This revelation powerfully challenges the long-standing dogma that rods and cones are fixed, clearly separated cellular identities. It suggests that the retina is far more malleable and adaptable than biologists ever gave it credit for. It proves that evolution operates much more like a computer hacker, mixing and matching open-source code to survive, rather than adhering to strict, unbendable biological blueprints. But the story gets even more fascinating when we look at the life cycle of these deep-sea mutants. The presence of these hybrid cells in the larval stage paints a remarkable picture of deep-sea life cycles and adaptation. The researchers found that these hybrid rod-like cones form extremely early in the larvae development. As larvae, these fish live closer to the surface, in that 20 to 200 meter range, where the intermediate light perfectly matches the unique capabilities of their hybrid eyes. But what happens when they grow up? As the lightfish and the lanternfish mature into adults, they undergo what is known as an ontogenetic shift. They migrate much deeper into the ocean, descending up to 1,000 meters into the bathypelagic zone, where the last vestiges of sunlight completely surrender to absolute, crushing darkness. In this pitch-black realm, intermediate twilight vision becomes entirely useless. Consequently, as these two species mature, their eyes undergo a radical transition, eventually adopting a traditional, rod-dominated visual system optimized purely for absolute darkness. However, the hatchetfish is a fascinating exception to this rule. Because the hatchetfish tends to remain in the mesopelagic twilight zone throughout its entire life, it actually retains these remarkable hybrid cells into adulthood. The presence of these hybrid cells in the larvae of all three species suggests a brilliant evolutionary stepping stone. The early appearance of these cells likely helps the larvae survive their highly vulnerable early weeks, and might even ease the complex biological and molecular transition into pure rods for the species that dive into the abyss later in life. It is a profoundly cool finding that shows biology absolutely does not fit neatly into human-made boxes. Scientists now strongly suspect that these hybrid cells might be much more common across all vertebrates, potentially including terrestrial species. In fact, traces of similar hybrid cellular structures have been previously hinted at in a few terrestrial reptiles, like certain types of twilight hunting lizards, but never with this astonishing level of direct genetic and molecular confirmation. The discovery of the hybrid eye cell is undeniably a massive triumph of pure biological science, but its implications stretch far beyond the realm of marine biology. We are currently living in an era of biomimicry, a rapidly expanding field where engineers and technologists look to nature's 3.8 billion years of research and development to solve complex human problems. The deep-sea hybrid photoreceptor offers an incredibly lucrative blueprint for the tech industry today. Currently, human technology heavily struggles with the exact same low-light limitations that plague standard vertebrate vision. Traditional night vision goggles or low-light camera sensors operate very similarly to biological rod cells. They heavily amplify faint light, but the resulting image is often grainy, artificially tinted, usually in bright green, and severely lacks crisp, dynamic resolution. Conversely, high-definition digital cameras, which act as our technological cones, require immense amounts of light or incredibly slow shutter speeds, which unfortunately cause severe motion blur just to capture a clear image. This marine finding is fascinating because it provides direct practical applications for technological advancement. In technology, creating digital sensors based on this unique cell structure could lead to dramatically more efficient cameras or goggles for low-light situations without ever sacrificing image sharpness. Imagine an entirely new generation of digital sensors built fundamentally on the rod-like cone architecture. A camera lens equipped with microscopic cylindrical photon traps that process data with the high-speed, high-resolution algorithms of a daylight sensor. This specific innovation could revolutionize everything from deep space astronomical imaging and advanced deep-sea exploration drones to consumer smartphone cameras and modern autonomous vehicle navigation systems. Self-driving cars currently rely heavily on optical sensors to correctly detect pedestrians and unexpected obstacles. A camera that can miraculously see with daytime clarity in deep twilight or heavy fog could quite literally save countless lives on the road. Yet, perhaps the most profound and globally impactful application of this deep-sea discovery lies directly in human medicine. The ocean is naturally an environment of extreme, unforgiving conditions. At a depth of just 1,000 meters, the hydrostatic pressure is roughly 100 times greater than at the surface. That is enough pressure to easily crush a standard military submarine. Yet, these tiny, fragile fish not only survive, but they seamlessly build and meticulously maintain highly complex cellular structures exactly like the retina under these crushing conditions. This extreme physiology has fiercely caught the attention of biomedical researchers, particularly those deeply studying human eye diseases. Glaucoma is a devastating and incredibly common human eye condition, currently affecting tens of millions of people worldwide. It is primarily characterized by an abnormal buildup of intraocular pressure, which is the fluid pressure inside the eye itself. Over time, this intense, unrelenting pressure damages the optic nerve and the delicate photoreceptor cells of the retina, inevitably leading to irreversible blindness. By intensely studying how deep-sea fish larvae construct and maintain their hybrid visual cells in the high-pressure environment of the ocean, scientists genuinely hope to uncover new, revolutionary biological resilience pathways. In modern medicine, learning precisely how these fish build this type of visual cell in the high-pressure environment of the deep ocean could essentially unlock new biological pathways highly relevant to human eye conditions such as glaucoma. If medical researchers can successfully isolate the exact genetic mechanisms or the specific structural proteins that protect the deep-sea fish optic nerves and photoreceptors from the absolute crushing weight of the ocean, it could pave the incredibly exciting way for revolutionary gene therapies or highly advanced neuroprotective drugs. Instead of merely attempting to lower eye pressure in glaucoma patients, which unfortunately is often insufficient to fully stop vision loss, doctors might eventually be able to fundamentally fortify human retinal cells, making them naturally resilient to pressure damage, much like the incredible eyes of the lanternfish. The discovery of hybrid eye cells in deep-sea fish larvae serves as a profound, humbling reminder of exactly how little we truly know about our own planet. The deep ocean is undeniably the largest habitat on Earth, yet it astonishingly remains less explored than the surface of the Moon or Mars. For 150 years, the smartest minds in biology confidently believed that the fundamental building blocks of vertebrate vision were locked into a strict, unyielding dichotomy. It took a half-centimeter fish swimming in the gloomy twilight of the Red Sea to beautifully prove us completely wrong. These brilliant findings loudly highlight the incredible elasticity and sheer genius of evolution. When directly faced with the seemingly impossible challenge of seeing in an environment that is neither truly dark nor fully light, nature simply did not just give up. It firmly refused to settle for the blurriness of rods or the blindness of cones. It hacked its own ancient code, seamlessly merging the hardware of one system with the software of another to thoughtfully create an elegant, highly effective hybrid solution. As humanity eagerly looks to the future, constantly seeking better cameras to accurately document our world, more advanced sensors to safely guide our autonomous machines, and novel, life-changing medical treatments to effectively cure debilitating diseases like glaucoma, we do not necessarily need to invent these complex solutions entirely from scratch. We simply need to look down. The biological ingenuity aggressively fueling the open ocean is overwhelmingly vast and largely untapped. The tiny hatchetfish, lightfish and lanternfish, peacefully swimming in the perpetual twilight with their microscopic rule-breaking eyes, are not just successfully surviving the dark. They are brightly shining a light on the absolutely boundless possibilities of biology, proving once and for all that nature's greatest, most revolutionary innovations are often silently hiding in the shadows, just patiently waiting to be seen. Thank you for listening to G Fun Facts Online.